appetite is regulated at different levels and by several organs such as the brain. The human brain is composed of diverse regions. One is called the hypothalamus, where we found a group of neurons involved in the hunger and satiety pathways. These neurons have the particularity to be in contact with blood circulation. The stomach, where the digestion takes place. And finally the gut. The gut epithelium layer is made of numerous enterocytes. Among them we find the enteroendocrine L cells. They can produce peptide hormones in response to diverse stimuli. The gut also contains a large number of bacteria and other microorganisms known as the microbiota. During food intake, L cells are stimulated by a reflex, and PYY for protein, YY, which is a satiety hormone, is released into the blood circulation as well as locally in the gut, where it stimulates the vagus nerve. When it arrives to the brain, the PYY leads to the inhibition of neurons implicated in the appetite pathway. We feel that it is time to stop eating. Activated L cells also produce glucagon-like peptide 1 or GLP-1, which stimulates the secretion of insulin by the pancreas. Insulin is known as the hormone, induces the capture of blood sugar after a meal. But it also goes further to activate the brain region that will produce the satiety hormone, the alpha-melanocyte stimulating hormone, or alpha-MSH. The alpha-MSH plays several roles. When it binds to the melanocortin receptors, it will activate satiety pathway both in the brain and via the blood circulation in the intestines, where it goes to activate the L cells and increases the production of satietogenic hormones, PYY and GLP-1. And what's about the microbiota? When we eat, nutrients also feed our gut bacteria. The number of bacteria increases and decreases along the day according to the meal rhythm. When bacteria grow, they reach a stationary phase during which they produce different kinds of molecules. Among them, one protein is called the CLP-B for casinolytic peptidase B. The CLP-B is a mimetic protein of the alpha-MSH satiety hormone. It works in the same way as a alpha-MSH in the gut, which means that the CLP-B will stimulate the L cells and increase the production of satietogenic hormones. Between meals, the CLP-B will pass in the bloodstream and activates hypothalamic neurons like the alpha-MSH does. The satiety feeling is prolonged, preventing us to nibble between the meals. Appetite regulation is a very complex mechanism driven by both hormones and bacterial signals in response to nutrients availability and metabolic status of both the host and its microbiota.